Sexual reproduction involves gamete formation and their fusion. Therefore, it is critical to know the structure of gametes. In this video, I am going to tell you the structure of sperm. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure that you subscribe my channel. So today we are here to look a very short and interesting topic, which is the structure of a sperm. So we have already talked about the process of spermatogenesis, and as well as we have seen the hormonal control of spermatogenesis. So in this is actually a short video in which we are going to look for the structure of the sperm. But before that, let's have a little brushing of what we already know from the previous videos. So in the video of spermatogenesis, we have discussed about that spermatogenesis is consisting of spermatocytogenesis and the spermiogenesis. So here, if you remember that spermiogenesis, basically it is conversion of a spermatids into the spermatozoa. So spermatids are something which are round cells, and the conversion of that cell into the spermatozoa, that process is called as a spermiogenesis. And this process is involving the differentiation of a spermatid into a highly differentiated, morphologically highly differentiated cell, which is called as a spermatozoa. So either we call it a spermatozoa or a sperm is basically a result of that process of spermiogenesis. So during that process, what is happening, that the organelles, they are highly compacted, their arrangement in such a way that they are arranged in a compacted form, as well as the next important thing which is important is that the cytoplasm is also, like, lot of cytoplasm is being removed during that process. So the sperm is having a very little amount of cytoplasm. And also the very important thing to remember is that the sperm is basically carrying the genetic material of a father, and it is carrying the genetic material to get fused with the ovum, which is having the genetic material of a mother. So this structure of a sperm is having a nucleus which is carrying the haploid DNA, and it is having the mobility. So it is mobile, it is moving. So basically its shape is such a way that is offering least impedance to its flow. So if it is flowing, there could be impedance by the secretions in the female genital tract. So that the shape is in such a way that it is that it is having a least impedance possible, and it could have a optimum velocity to reach to the ovum. So with this, we can start talking about the sperm or the spermatozoa. As I already said, that the spermatids are something which are round epithelial cells, and here they are differentiated into a very highly differentiated morphological cell called as a sperm or the spermatozoa, which is having head, middle piece and tail. So you can see it is basically a long cell, elongated cells. The organelles are being compacted. Okay, so the shape is such in such a way which is offering least impedance to its flow. So the structure is actually facilitating its function. So you can see it is having three parts: head, midpiece and tail. So this is the head, this portion is head, and this portion is midpiece or middle piece, and this portion is tail. So the head is having, you can see here, the head is having the acrosome, or also called as an acrosomal cap, because as it appears to be capping the nucleus, also called as an acrosomal vesicle. Then the nucleus is being there, and in the middle piece the centriole is there, mitochondria are there, and the tail is having the flagella, which is taking or moving it in forward direction. So one by one we are going to deal in detail with the components of each of these parts. So first we talk about so all these structures are being enclosed by a plasma membrane that is called as a sperm membrane. So first we talk about head and the structures which are being there in the head. So acrosome or the acrosomal cap is basically a structure which is being derived from the Golgi apparatus, and it is containing hydrolytic enzymes which are important for causing the degradation of the proteins or the matrix surrounding the ovum, and it is facilitating the fertilization by causing their degradation. So you know that every structure of the sperm is somehow facilitating its function. So acrosome is actually facilitating the cortical reaction and the acrosome reaction, so that finally the fertilization could take place. Then after that there is nucleus, which is having haploid content. So we already know that the gamete is being produced as a result of meiosis, so it will have the half the number of the chromosomes as that of a somatic cells or a germ cell. So it is having haploid, that is one copy of single chromosome, and DNA content is 1C. So here very important thing, which is like as I already said, that during the process of the spermiogenesis, what is happening, that the cytoplasm is being decreased, is being lost, minimum cytoplasm is being maintained, and the organelles, they are also being compacted. All of these is being done to decrease the volume of the cell. So basically here also the DNA packaging is there in the nucleus, and DNA is extremely tightly packaged by using the primarily using like 95 or 85% of DNA packaging here is being done by the some non-histone proteins, which are somehow like histones only. These are also positively charged, rich in basic amino acids such as lysine and arginine, called as a proteamines. So these are involved in densely packing the DNA, and this finally leads to decrease in the volume of the nucleus. And as well as because DNA is so tightly packed, always remember that there is no transcription which is happening in the sperm. The sperm is transcriptionally silent. So that is about the head. Now we can talk about the midpiece. So midpiece, as you can see here, is having lot of mitochondria which are spirally arranged. So this is a mitochondrial sheath which is there. So as we know that mitochondria is the storehouse, or sorry, not the storehouse, but the energy house of the cell, which is being producing ATP. So the same thing is being done here also. It is involved in the oxidative phosphorylation. By using the electron transport chain, it is producing the ATP, and that ATP is being used by the sperm motility apparatus to propel the sperm in a forward direction. So the major fuel for the sperm is the fructose. So here is an important thing which is coming, that we are saying that mitochondria is producing ATP. So how does the mitochondria is producing ATP? It must be requiring some fuel, some substrate must be there, which is going to undergo the metabolism, and then the ATP is being derived by breaking that fuel. So that is being done by the that is the fuel is the fructose. So this is just a very short overview, because like I already said, this is a short video, so it is not possible to cover many things in detail. So here you can see that this is the acrosome, this is the nucleus, and this is the middle piece which is having the mitochondria, and this is a portion which is in between the head and the middle piece. So you can see that these are the this is the plasma membrane of the sperm, and they are having the transporters. So these glutes are the glucose transporters. You must be knowing aware about them. So they are actually leading to the entry of the sugars inside the cell. That is majorly it is fructose, because fructose is a major fuel which is being present in the semen produced by the seminal vesicle. So majorly it is being used, the major fuel which is being used by the sperm is the fructose, but sometimes it can use glucose also. So you can see here that this can enter, like if this is the semen and this is having the fructose, from that by the help of the glutes on the plasma membrane, it can enter inside the cell, and then it can undergo the process of glycolysis in the cytoplasm. So even though the sperm is having a very little cytoplasm, it is still containing some cytoplasm and also the enzymes of the glycolysis, so that it could be converted into the pyruvate and then acetyl coenzyme A, and then it is entering in the mitochondria mitochondria, and then it is in the mitochondria, it is undergoing the tricarboxylic acid cycle or the Krebs cycle, and then finally oxidative phosphorylation to produce the ATP. So all this shows that there is an aerobic metabolism of the fructose which is happening, which is done by the mitochondria, and so much mitochondria is being there in the sperm. So you can see that almost all the organelles, they are being removed from the sperm. Golgi, a derivative of a Golgi body is being there, which is the acrosomal vesicle, and the other organelle which is being there is the mitochondria, and it is being there in a huge amount because the sperm is requiring ATP to move forward. For its motility, for its mobility, the ATP is required. And how does the ATP is being produced? By metabolizing the fructose, that is a major fuel source present in the seminal present in the semen produced by the seminal vesicle. So now we can see the other things, or we can simply say that let's talk about the sperm motility apparatus. How does the sperm moves? So it's very simple. If we know about general physiology, that the motility of the sperm is the mobility of the sperm or any cell is by the cytoskeleton. So here, which type of cytoskeleton is being involved in moving the sperm? The answer is microtubule. And which molecular motor is being involved? The answer is dynein. So this molecular motor and this microtubule, both of them are actually leading to the movement of the sperm, and dynein is an ATPase which is going to utilize the ATP produced by the mitochondria, and that ATP is going, which is a chemical energy, will be converted into the mechanical energy by this molecular motor dynein, and thus the sperm will move forward. So you can see that there is a centriole. So here in this diagram, okay, this is a centriole. So centriole is something which is also called as a microtubule organizing center. So basically this centriole is having a pair of centrioles from which many microtubules are being arising. So this is a negative end of the microtubules, and the positive ends are something which are pointing or moving away from the microtubule organizing center. So this is the cytoskeleton microtubule, which is basically having a polymer of a tubulin protein. So this is the centriole, which is located here in the middle piece only, and from there, if you see these lines, these are the microtubules. Okay, so now if we have a cross section of this, if we take a cross section of this part is called as an axoneme. So this is the axoneme. So if this is the microtubule organizing center and microtubule is arising from that, so if we take the cross section of this is how the arrangement of a microtubules in the core of axoneme is being there, which the microtubules which are emitting from the centriole. So you can see here there are about 2 microtubules which are in the center. So this is basically a 9+2 arrangement which is being there, 9 doublets on the outer and 2 doublets inside. Okay, 9 doublets outside and 2 singlets microtubules inside or in the inner one. So this is the axoneme, which is the kind of a cytoskeleton arrangement present in the flagella, and the molecular motor is already, I told you, is a dynein. So it is going to attach to the microtubule, and it is going the bending of the flagella or the axoneme in either direction, leading to the propulsion in the forward direction. So tail is actually having the extension. So you can see that from here the microtubule organizing center, the microtubules are arising from here and they are going up to here. So tail is having the axoneme, and the mechanism, I already told you that there is a molecular motor which is using the ATP, converting the chemical energy into mechanical energy, leading to the movement of the sperm in the forward direction. So we have talked about the structure of the sperm, that it is having a head which is having acrosome and nucleus. It is having a midpiece which is having mitochondria, centriole, axoneme, and it is having a tail which is actually an extension of the axoneme which has started in the middle piece only. So the important thing is that the sperm has lost all the cytoplasm, almost all the cytoplasm, and it is being made in a shape which is having least volume. The compaction of the organ is being done. So with this, we are at the end of this video. These are the main references of this video, and apart from this also I have referred boron and guyton. So but these references are the main. So if you have any doubt, you can look into these references. So if you like this video, please like, share and subscribe our channel. Comment down your suggestions and doubts. Till then, keep learning.